Hello, I'm Sina Faizi, a PhD student in computer engineering at the University of California, Irvine. I work in the Advanced Cyber Physical System Laboratory under the supervision of Professor Muhammad Al-Farouk. In this video, I'm going to present a novel side channel attack methodology against commercially available DNA synthesizers. This work is a result of a collaboration between UC Irvine and UC Riverside, and it's supported by the National Science Foundation in an effort to manufacture secure high-throughput DNA synthesizers. The biological information of all living organisms are stored and transferred in the form of DNA molecules. These molecules are chains of the four base types, A, G, C, and T, which are attached together to form the well-known double helix that you can see here. Different combination of the bases in DNA sequences translate to different proteins, each with different function in a given organism. But DNA sequences are not just produced in nature anymore. Over the last century, automated DNA synthesizers have emerged which can cost-effectively produce custom synthetic DNA molecules. Currently, the field of synthetic DNA security is primarily focused on one aspect of custom DNA synthesis, and that's bioterrorism. The fact is that some synthetic DNA sequences can be used to produce deadly diseases which can result in the extinction of human beings. For years, government agencies have tried to nullify this threat with various regulations, monitoring systems, and training. However, there is another aspect of synthetic DNA technology which needs more attention from security experts. In recent years, many companies have a vested interest in creating synthetic DNA molecules. Scientists have started to use custom DNA molecules to produce life-saving drugs, better medical treatment procedures, and various optimized crops to feed the growing population of the Earth. Recently, researchers have even started to investigate the possibility of using DNA molecules as an ideal medium for data storage. In almost any of these applications, a huge financial investment is required to engineer the order of the bases in the synthetic DNA molecule to give them a certain functionality. In other terms, synthetic DNA sequences are intellectual properties and they need to be protected against possible adversaries. In this video, I'm going to introduce a new way of stealing the order of the bases in the DNA synthesis process. We show that it's possible to take advantage of the acoustic side channel to predict the structure of the synthesized DNA molecules. In order to attack a target DNA synthesizer, we propose that an attacker can simply place a microphone in close proximity to the DNA synthesizer and record the acoustic noise while the machine produces DNA sequences. In this scenario, the attacker can be a disgruntled employed, a visitor who leaves his phone around the machine, or even a compromised electronic device which might be present in the environment that the DNA synthesizer works. Once the attacker collects the recorded noise, he will use his pre-trained estimation functions to predict the order of the bases in the synthesized DNA sequence and sell it for a profit to a competing company. First, let's take a look at how DNA synthesizers work. The commonly used DNA synthesizers carry out four repetitive steps sequentially to produce the output DNA molecules. As you are going to hear next, the machine generates a continuous noise with some clicking sounds once in a while. Basically, the continuous noise is from the fluids running in the system's pipes, and opening and closing of the solenoid valves causes click-like peaks in the collected signal. The chemical synthesis process can be easily tracked and mapped to the noise that we hear based on the user manual of the machine, and the peaks will help us to segment the signal to the base delivery segments. Once we do the mapping, the only challenge left is determining the type of the delivered base. We use state-of-the-art machine learning and signal processing techniques to classify the type of each base. Next, I'm going to present to you our experimental result. We choose the applied biosystem DNA synthesizers to carry out our experiments. According to the biomedical equipment sales company, this is one of the most widely used DNA synthesizers in the industry. We randomly placed our recorder in less than 10 centimeters proximity of the DNA synthesizer and recorded the noise that the machine generates. The microphone that we used was a Zoom H6, but there is nothing special about it, except the fact that the most of its characteristics are available online. However, a commonly used cell phone could be also used for recording. Here we show that it is possible to train the classifiers with a limited number of training samples. In this graph, the x-axis shows number of samples used for training, and the y-axis represents the accuracy of the classifiers, which is basically our total accuracy. We use various types of classification methods available in literature, such as neural networks and random forests, to make sure that we are utilizing the best possible classifier. As you can see, most of the classifiers start to converge after approximately 180 training samples, and hence not too many training samples are required. On average, our results shows that the classifiers that we use can reach 88% accuracy in predicting the type of each particular delivery. We achieved good accuracy for predicting each random base, but to make sure that the attack model will work in a real-world scenario, we crafted a new experiment. We asked one of our collaborators to synthesize a meaningful DNA sequence without giving us any information. We put a recorder near the machine and collected the acoustic noise for 1 day. The next day, we collected the recorded sound and fed it to our trained model. We then emailed the predicted sequence to our collaborator to compare it with the original sequence. Here you can see his response email. As you can see, our attack model had only 6 missed predictions out of 45 bases, which matches the attack methodology's accuracy calculated before. We carried out the same procedure for 3 other interesting sequences, and our results accuracy was consistent for every sequence. Now there is only one question left. Is 88% accuracy on average good enough? Actually, the answer is yes. The next step after single base type prediction is application-specific post-processing. For instance, here we knew that the reconstructed sequence has biomedical use, so we fed it the predictions to a publicly available tool called BLAST. And it gave us almost same result as if it was using the original DNA sequence, which basically means that we were able to perfectly steal our target DNA sequence. Thank you very much for your attention. I hope you enjoyed this video. For further details on the implementation of the attack methodology, you can refer to our latest paper, which is published in NDSS 2019.